Have you ever stopped to think about the journey your food takes before it lands on your plate? It's a long and winding road, my friends, and along that road, things can happen. Good things like proper aging of a cheese, and not so good things like unwanted microbial gatecrashers or a sneaky bit of something that shouldn't be there. That's where food testing comes in. It's not just about rules and regulations, it's about trust. It's the silent, scientific promise that what you're about to eat is not only delicious but more importantly safe. Think of it as a quality control system for your very own fuel source, ensuring every bite is what it claims to be. This whole process is a bit like being a food detective. These scientists are culinary sleuths, use a fascinating array of tools and techniques to interrogate food at a microscopic level. They are looking for clues. Is the texture right? Does it have the correct nutrients? Are there any microscopic villains lurking within? These questions are vital. The answers protect us from getting sick, ensure we get the nutrition we pay for, and maintain the quality we expect from our favorite brands. Without this rigorous examination, our food system would be like the Wild West, unpredictable and potentially hazardous to our health. Now you might be thinking, Alton, this sounds complicated. And you're not wrong. The science can be deep, but the idea behind it is simple. We need to verify what's in our food. We check for the good stuff, like vitamins and proteins. We hunt for the bad stuff, like harmful bacteria or undeclared allergens. And we confirm the physical stuff, like whether your potato chips have the right crunch or if your ice cream is perfectly smooth. Each test tells a small part of a larger story, the biography of your food, from the farm to your fork. So why should you care? Because knowledge is power, and understanding how your food is kept safe empowers you as a consumer. It helps you appreciate the price of quality and the unseen efforts that go into every carton of milk, every loaf of bread, and every single strawberry. It's the invisible shield that guards our global pantry, ensuring that when you sit down for a meal, your only concern is whether or not you want a second helping. And let's be honest, you probably do. This elaborate system of checks and balances is the bedrock of a safe and reliable food supply. Let's start with the basics. Physical analysis. This is all about the tangible, measurable properties of food. Think texture, viscosity, density, and color. It's the science of mouthfeel. For example, when you bite into a crisp apple, that satisfying snap is a physical property. Food scientists use machines like texturometers to measure the exact force needed to puncture that apple's skin. Or they might use a viscometer to ensure your favorite brand of ketchup flows from the bottle just right. Not too fast, not too slow. This type of testing ensures consistency, so the bag of chips you buy today has the same delightful crunch as the one you bought last month. Next up, we have chemical analysis. This is where we look at the chemical components of food. We're talking about the big stuff like fats, proteins, and carbohydrates, but also the smaller things like vitamins, preservatives, and pH levels. A classic example is the Kjeldahl method, a time-tested process used to determine the protein content in a food item like a power bar or a cut of meat. By measuring the amount of nitrogen, scientists can calculate the protein. This analysis is crucial for creating those nutrition labels we all depend on to make informed dietary choices, ensuring the label's claims are actually true. Then we dive even deeper with molecular analysis. This is where things get really high-tech, right down to the DNA level. This technique is the bloodhound of food testing, capable of sniffing out specific genetic fingerprints. For instance, if a product is labeled as 100% beef, molecular analysis, often using a technique called PCR, polymerase chain reaction, can confirm it. It can detect if cheaper meats like horse or pork have been mixed in. This same technology is used to identify genetically modified organisms, or GMOs, or to pinpoint the presence of specific allergens, like peanuts or soy, that could be dangerous for some people. Finally, we have elemental and microbial analysis. Elemental analysis uses powerful instruments to find and measure specific elements, like minerals or, unfortunately, heavy metals. A machine called an atomic absorption spectrometer can detect trace amounts of lead or mercury in fish, which is vital for public safety. On the other hand, microbial analysis is the hunt for living organisms, bacteria, yeasts, and molds. Scientists take a food sample, place it in a petri dish with a growth medium, and wait. If nasty bugs like Salmonella or E. coli are present, they will multiply and become visible, signaling a major safety hazard that needs immediate attention. At first glance, these testing methods might seem like completely separate worlds. One pokes food with a machine to check its crunch, while another maps its very DNA. However, they are more connected than you might think. The common thread weaving them all together is the pursuit of data. Every single technique, whether it's measuring the viscosity of honey or sequencing the DNA of a strawberry, is designed to gather specific, quantifiable information. They all aim to answer the fundamental question, what exactly is this food made of? They are all tools for verification, for holding food to a standard. The differences, of course, are what make them uniquely powerful. The main distinction lies in what they are looking for. Physical analysis is about structure and form. Chemical analysis is about composition and nutrients. Molecular analysis is about genetic identity and allergens. Elemental analysis is about atomic building blocks and contaminants. And microbial analysis is about living contaminants. You wouldn't use a texturometer to find Salmonella, and you wouldn't use a Petri dish to measure protein content. Each tool is specialized for its own unique task, like using a different knife for butchering, filleting, or paring. Consider the case of a simple jar of peanut butter. Physical analysis would ensure it has the perfect spreadable consistency. Chemical analysis would verify the protein and fat content listed on the nutrition label. Molecular analysis could confirm that only peanuts were used, ruling out other undeclared nuts which could pose an allergen risk. Elemental analysis might check for trace heavy metals from the soil where the peanuts were grown. And finally, microbial analysis is absolutely critical to ensure the batch is free from Salmonella, a known risk in peanut products. Each test provides a different layer of safety and quality assurance. This highlights their complementary nature. One method's weakness is another's strength. Chemical analysis can tell you the protein is there, but molecular analysis can tell you if that protein came from the source it was supposed to. Physical analysis can tell you if a fish fillet is firm, but only elemental analysis can tell you if it's high in mercury. No single technique can provide a complete picture of a food's quality and safety. They are not in competition with one another. They are members of a team working in concert to build a comprehensive profile of the food in question. So, why do we need this whole orchestra of techniques? Why not just pick one and call it a day? The answer is simple. Food is incredibly complex. A single food item is a mixture of physical structures, chemical compounds, biological materials, and sometimes unwanted guests. Relying on only one method would be like trying to understand a city by only looking at a road map. You'd see the streets, but you'd miss the people, the buildings, the culture, and the economy. To truly understand the city or the food, you need to look at it from every possible angle. Think about it this way. Ensuring food safety is about managing different types of risk. The risk of a food having a disappointing texture is a quality issue handled by physical tests. The risk of it not providing the advertised nutrition is a chemical issue addressed by chemical analysis. The risk of fraudulent labeling or hidden allergens is a molecular issue. The risk of contamination with invisible toxins like lead is an elemental issue. And the risk of it making you violently ill is a microbial issue. Each risk requires a specific defense, a specific test to keep it in check. This multi-pronged approach is the foundation of our modern food safety system. It creates a net with very fine holes, catching potential problems that a single, broader method might miss. For example, a steak could pass a microbial test with flying colors, but molecular analysis might reveal it's not beef at all. A salad mix might have the perfect vitamin content according to chemical tests, but still be recalled due to an E. coli outbreak discovered through microbial testing. The layers of testing work together to ensure that by the time food reaches us, it has been thoroughly vetted from multiple perspectives. Ultimately, this symphony of science is what allows us to eat with confidence. It's a testament to how far we've come in understanding what we consume. From checking the crispness of a cracker to hunting for the DNA of a pathogen, these diverse analytical techniques are the unsung heroes of the kitchen. They are the guardians of our grocery aisles and the gatekeepers of our health. They ensure that the food we rely on to nourish our bodies is safe, authentic, and wholesome. And that, my friends, is a recipe for a very good eat indeed.